I would like to thank Srijit once again for inviting me. In several occasions, he used to call me for the talk in this Kerala University, and thank you Srijit again for inviting me. It's like little bit different as you are almost 6 days come across about the wide microscopic techniques, especially the imaging. It's like quite different types as far as fluorescence microscopy. Raman microscopy is one of the leading technology in the current arena because of its huge applications, especially in the biomedical applications, especially in the bioimaging and the cancer diagnosis. So, I will talk about around 45 to 50 minutes about the basic discovery of Raman microscopy, Raman scattering especially, and how it's implicated its application towards the bioimaging, especially in the detection process in different areas, not only in the cancer, in wide field of applications. So, the outline of my talk is the introduction, how we can come about the surface enhanced Raman spectroscopy and its application, especially in the biomedical, and current progress on diagnosis and therapy, and how we can make a probe out of it, and which can be translated and for the clinical translational research for together theranostics, including therapy and diagnosis, and definitely its future perspectives. So, before I go is some very few limited part of it is scattered in a different energy of the incident photons, which is called inelastic scattering or Raman scattering. So, this finest discovery made by Sir C.V. Raman in 1928, and he was so confident about his discovery that he think about I am going to get Nobel Prize very soon, and within 2 years 1930 he got the Nobel Prize for this discovery. And from that I just give you some of the basic things, as I said that when this is energy is almost equivalent with that of the incident energy, which is called the Rayleigh scattering, which is elastic, and the Raman scattering, which is inelastic, where you can get the different energy compared to that of the incident photons, and which is two different modes, which is called Stokes and anti-Stokes Raman scattering. So, this is very basic principles of the Raman scattering which they have discovered. But if you way back to the history of this discovery from 1923 to come about 1997, where Raman and Krishnan, when they have first discovered it is a feeble fluorescence intensity and it can be a inelastic scattering of light. Then after his Nobel Prize, this is a huge gap around more than 50 years, around 50 years or less close to. Then people are working mostly on materials and molecules about its phenomena in this area. But in 1997, Felsman, who first come about a new idea that when you are doing this Raman scattering and in a electrode, then he incorporates some way the pyridine molecule on it, and he got a huge enhancement of the signal intensity from the pyridine come about, and this idea come across, and then he think about it is a really a surface enhanced process, and which come about when the surface phenomena of the metal substrate is very crucial. So, he first discovered that surface enhanced Raman scattering, and then out of this gap people are working many ways of the surface enhanced Raman scattering in materials and molecules, and even in 1997 and onwards people are capable for detection of the single molecule out of it. So, from 2003 to now onwards, if you go about the surface enhanced Raman scattering history, then people tuned it in different applications, especially the biomedical applications. So, now this field totally utilized for detections in many areas which I will come across, and I am talking about mostly what is the invention in the biomedical application in this field. So, just one example, you know the cyclohexane, the solvent, which if you irradiate, if you use the laser beam at 488, and you get in the left side you get very good visible fluorescence, and this is mostly from the Rayleigh scattering, which you can come across. And this Rayleigh scattering, but if you somehow if you filter off this Rayleigh scattering, you may get this one in the right hand side, and which is called the Raman wavelength, which is a longer wavelength and Raman scattering. So, you may filter off the Rayleigh scattering, you may get the Raman scattering. So, this is one just short examples how it come out. So, now I will give you some very few examples and the basic principles how it is unique in a way compared to the infrared spectroscopy, IR spectroscopy. IR and the Raman are the complementary to each other, but the thing is that we may distinguish why it is Raman, why it is the molecule is IR. The basic principles that the molecules, if you look on the molecules, what its position, it is a symmetric molecules or it is a asymmetric molecules. So, if it is a symmetric molecules, you may get mostly the Raman phenomena, but if it is a asymmetric molecules, you may not get Raman phenomena, it comes in the IR. Another way I can explain you that if a molecule having permanent dipole moment, then the wavelength, scattered wavelength, what you got it, the phenomena out of it from the permanent dipole moment, it is coming in the infrared. But the Raman phenomena is not the change in the permanent dipole moment, it is rather the polarizability of the molecules which came out of these molecules, and then it will be coming out as a Raman. So, if you see the carbon dioxide, which is a symmetric molecules, but if you think about it can be changed its polarizability not in the permanent dipole moment. So, it is a Raman active rather IR inactive. So, if you make a tuned molecules which is not centrosymmetric or then you have to generate in its permanent dipole moment, out of which then you can generate the IR. So, this is the very basic principles. People are getting confused sometimes why it is, which molecule will be the coming in the Raman and which molecule can come in the IR. So, I will give you some of the basic advantage of the Raman compared to the IR, and why it is used in so much applications, especially in the biomedical. Because water you can use as a solvent in the Raman, one good way where you can do the most of the biological samples in the native state, and you can work out through it. And another things that although the Raman spectra results the molecular vibration at the IR frequencies, but the spectrum is obtained in the visible light or in the near IR radiation, that is one of the biggest advantage over IR. And totally symmetric vibrations we can observe from out of it, and its intensities definitely is proportional to the concentration of the laser power, that also it is one of the good phenomena. These are the few basic advantages, there are lots of advantages is there over the IR, but the main disadvantage in the Raman spectroscopy is that it is very weak. You need high amount of laser power to generate the corresponding spectra out of it. So, that is the one of the biggest disadvantage you can say, and out of 10 to the power 6 photon, one photon maybe you can implicate in a Raman photon. So, when it comes in the SERS phenomena, that is that the phenomena you have the metallic substrate, if you incorporate the molecules of interest which you want to check as in Raman mode, then if you put on the laser you get the enhanced signals. Why? I will tell you the explanation some extent on it, but this enhancement you may translate to up to 10 to the power 10 to 14 fold in a way, and this is the enhancement coming out from the surface enhanced Raman. There are two types of surface enhanced Raman you may utilize. One is called the level free SERS, surface enhanced, where your substrate is the metallic nanoparticle, either you used any metallic nanoparticle like gold, silver, platinum, any way, and the analyte of interest you want to detect, that you can incorporate in it, and due to the close proximity of the metallic surface and your analyte, two phenomena is happening. One is the electromagnetic enhancement and is the chemical enhancement, and out of it you can get the corresponding molecular vibration change in the molecule of the analyte, and you get the Raman spectra. That is the level free, directly you can use it. Another is the with level detection, where or it is called the targeted Raman, targeted SERS, where you may not get a very prominent signal or prominent peaks from the analyte itself. In that case, you can incorporate a Raman active molecules on the surface, and this used as a signature molecule for it, and with this signature molecule you can determine the analytes you want to detect. That is, you may incorporate and you can get the Raman signals. So, mostly people are working into these methodologies, either level free or in a direct targeted fashion. So, Sluker first, he is in his paper in the physical chemistry in 2009, he established this phenomena, and also it is translated by NIIST, who is one of the pioneer in this field, and he wrote this review on it. So, as I told you, the two phenomena is working. One is the plasmonic phenomena through the electromagnetic enhancement translated, and that caused the huge enhancement, and another is the chemical ionizations mode, where your analyte molecules may form a covalent or non-covalent bond. If it is a covalent bond, it is called the chemisorption, and if it is a non-covalent bond, it is called the physisorption, and out of which the molecular energy level or molecular orbital, if you comes across it, the highest occupied molecular orbital and the lowest occupied molecular orbital, you may tune it and you get the closest band gap, and you may tune it the enhancement through your chemical enhancement way. So, these are the two theoretical aspects people are believing to get this enhancement. So, this is one it. Now, I will show you some of the basic advantage and applications. One is the you may use it, this phenomena, to detect the pigments. There is lots of applications is there, I will show you, but this is one of it, that you may detect some of the pigment molecules and from either plant or any other materials, and you may get the specific molecules, specific inelastic scattering of the molecules that can be very unique, because its bandwidths are very sharp. So, you may easily detect the molecules, and you may that detection will be a signature peak for that particular identifications. So, that way you may detect as a level free detection, the identification of the pigments is one case. But when you translate in the biomedical applications, you need to tune many things. One is that this is your substrate, which is a gold probably nanoparticle, is a Raman active substrate. Then in the targeted fashion, you may incorporate some of the Raman active molecules to detect, and you have to functionalize many biomolecules. For that, you have to do some of the functionalizations in surface coatings or surface functionalizations, and that functional groups will be tagged with many of the targeting molecules, either small molecular proteins or any peptides or any other nucleic acids that can be used for detect the specific analytes. So, this is the surface modification is one of the way you may improve the stability, other way you may improve the stability of the molecules, and the direct fashion you can detect. And also with this you may conjugate even many of the targeting molecules, either antibodies, and even it can be tuned in many proteins. So, this is one of the biggest advantage where you can use it in biomedical applications. So, now I will tell about some of the substrates which nowadays people are using in surface enhanced Raman. The most common one is the gold nanoparticles people are using, because it has a unique plasmon peak and that can be tuned in different wavelength region from visible to NIR. So, gold nanoparticle can be used, and upon its size and shape also it can be tuned in the wavelength region. That can be used as a good substrate. Sometimes people are using the nano cell, where it is a cavity, the nano cells having the surface, and where maybe you can tune in a way that you utilize many of the biomolecules together in it, and which having different property compared to the gold nanoparticles or spherical nanoparticles. Sometimes people are using rods which having two edges, and it has different aspect ratio. Aspect ratio means its length and the width, that ratio is called the aspect ratio. With that ratio you may tune different physical property on it. So, these are the common substrates you can utilize. Apart from that, nowadays people are using different size and shape molecules like nanostar, which is used, nano flowers people are used, and also gold and silver are the biometallic nanoparticles. Actually, among gold and silver, silver is the much better substrate as per the enhancement. So, people are using gold core silver cell as a Raman substrate. So, these are the quite useful substrates people are using. Now, upon this substrate, you may think about many of the bioconjugations on it. This is a spherical particle, or you may use as a chip, where you can coat that gold plate and you may conjugate the antibody, and you can detect the corresponding analytes through sandwich approach, primary and secondary antibody conjugations. And also the different analytes, like you may use it for this detection modality. So, these are the principles people are using nowadays, and I will show you several examples and finest discovery in the very recent years. So, coming to the application side, first people are started with the toxic chemical detection, chemical warfare, and then people are used to detect the dyes and other materials. Even in the chemical reactions, checking their oxidation state people are using, and now people are using mostly in the biomedical applications detections. Even in the airport, custom peoples are nowadays start using this hands on Raman to check the some of the foreign materials and toxic identities through that area. So, that covers a lot, but in I am going to talk about mostly the biomedical applications, where it can be utilized in many ways, in either in the detection mode of in the field of cancer, in the field of other disease, and also in even in the environmental pollution states. So, if you this is 2000 up to 11 approach, if you see that how this field is growing up and exponentially with terms of publications, patents, and also the discovery, and some of the things are also in the form of prototype clinical application stage, hoping it will come up in the modality soon. So, I will give you some of the very nice examples in the field of cancer treatment and diagnosis. One of the therapeutic treatment modality is the photothermal therapy, where in the nanomaterial, if you generate heat, if you generate the laser, if you irradiate the laser, then on that nanomaterial you can generate heat, and that temperature generally it will be in the 40 to 50 degree Celsius temperature. If you use that localized temperature in the cancer cells targeted way, then you can kill the cells, cancer cells, and that is called the photothermal therapy. So, here one of the finest example, they are using the gold nanocube, where they decorated with some of the targeting group like cyclic RGD, and that they can use to kill the cancer cells targeted fashions. But another way they have done the the cell division, in cell division is a spontaneous process, and they have called this is a biomedical snapshot, where you may find out the normal cell division and the cancer cell division, how it can be reflected. You find out in the cancer cell division, the thing is that the beta sheet change is very much abundant, whereas in the healthy normal cells it is not so much abundant. So, they have checked the specific cell division arena in terms of this Raman scattering, especially the surface enhanced Raman scattering. How it is? So, these are the substrate, this is the TEM image, and this is called the dark field image of that substrate. And especially when you go to the cell division, starting from the prophase, metaphase, telophase, anaphase, what happened in the real time, that they translate in the Raman scattering. There are three or four changes which is very significant during that cell division. One is that the SS vibration, CS vibration, and one of the amino acids, especially the phenylalanine or tyrosine, there will be a change in its abundance and the amide bonds abundance. That all has been reflected, means when this division occurs, the abundance of the phenylalanine will be quite increased, that they have shown in this pattern. So, these changes in real time can be monitored, and that can be translated in the form of Raman. You may do it in real time fluorescence microscopy, but it also be translated in Raman. So, this is one of the recent work, and there is one very recent work which is I say it is one of the fantastic things they have done, that is also a photothermal therapy. And in that photothermal therapy, what happened, they can translate the Raman spectroscopy or surface enhanced Raman scattering in terms of metabolomics and proteomics. How? In the metabolomics, if you think about the specific amino acids and its derivatives, how it will be correlated in the Raman, its abundance. Also in the proteomics, when you find out the pathway and its apoptotic things, that can be translated. So, they are just taking one amino acids, that is called the phenylalanine, and it is associated with the phenylalanine. So, what they did, what they found, that in that particular change, when you induce the photothermal therapy and induce the apoptosis, the phenylalanine abundance is quite increased, apart from the other spectral features, mostly in the DNA backbone cleavage and the lipid or amino acids, other amino acid changes. So, they correspond that the phenylalanine abundance, if you can check it, that will be reflected in the even in the proteomics and the metabolomics. They have done lot of the proteomics analysis, mostly done by the array of the mass analysis, and that particular fragment they got the phenylalanine abundance, which is reflected in the Raman. So, this is Al-Said, which is one of the pioneer in this field in Emory University in Georgia. So, he is published in 2016 this work, and he has done extensively in this field. One of my research area is the drug delivery. So, this is one of the finest work I just gone through, that is that the doxorubicin, which is used as a potent chemotherapy, and when you induce the apoptosis through this drug, how you can check whether it is internalized, whether it is in the right target site, that can be translated in both ways. One is the fluorescence, another is the Raman. Fluorescence, as it is a fluorescence molecule, doxo is a fluorescent molecule, so its emission is around 530, but the way we can check it as a turn off and turn on mechanisms, where the gold nanoparticle is conjugated with the doxorubicin through hydrogen linkage, and this whole construct will be when you used or incubated in the cancer cells, it will be endocytosed and it comes to the target site, and in presence of the lysosomal pH, this hydrogen bond will be clipped and this molecule, this doxo molecule will be free, then its fluorescence will be turned on. On the other hand, when it is conjugated with the RAM with the SERS substrate, its fluorescence is quenched, you get only the Raman peak on it. So, this is translated here. This side is the Raman scattering, surface Raman scattering, and this is the fluorescence modality. So, when it is intact with the gold nanoparticle, this is here, you can see the Raman bands on it, and when it slowly goes in the cells, this is the live cell experiment. So, when it goes to the cells, slowly this hydrogen bond is clipped, and you see the slowly the peaks 460, which is mostly the SS linkage, is diminished and the spectra is flattened slowly. Whereas, once it goes in the cells, upon time you can get the enhanced fluorescence. So, the doxo is released. So, this one of the finest work to show that these two modality can be correlated with turn on and turn off mechanism. There are quite a few examples, maybe I will go through a little quickly, because these are the finest discovery in recent 1 and 2 years time. This is little old work, but here they used a gold nano popcorn, like that is a nano cube, and it is surface functionalized, including the target molecules. Is rhodamin 6G is a Raman reporter they incubated in the RNA aptamer, and this is conjugated on it, and this is targeted to the prostate cancer. That is why they used the prostate specific antigen on the nano construct. So, this is the whole construct, and when they irradiate with the laser to induce the photothermal therapy, what happen? The molecule will be disassembled, and once it is disassembled, then the thing is that it will be like that. Once it is slowly disassembling, then this Raman band will be disappeared, because this rhodamin 6G not in intact with the nanorod. So, that has been reflected. So, there are many characterizations ways in the physical mode they have checked with the target specific, because it is a specific to the prostate cancer cells. They use some of the specific prostate cancer cells, and as a negative control different cell lines they have used, and they showed that it is a very target specific, and it showed that when it will be delivered as a photothermal therapy, then it can be translated through the Raman. There are another finest examples in this area, where you may, when you induce the apoptosis, then what happen? It is a nuclear targeting and the DNA cleavage is happening, and if you see that DNA fragmentations, that can be nicely translated and in this process during the apoptosis. So, this is the protein denaturations, and in this stage you find that this native stage of the protein and this is the denatured stage of the protein. So, when it is denatured stage, you may find that its intensity will be reflected. It is the DNA fragmentation stage in this apoptosis. In the DNA fragmentation stage, here you may find that DNA phosphodiester linkage is around 830 centimeter inverse, which is reflected here. When it is fragmented, this band will be disappeared, that is here. This is the protein degradation stage. In the protein degradation stage, you may find the amide bond in the range of 1300-400 regions. When it is degraded, that band is disappeared, and that can be translated in cell even, and that they have done. There are many ways. Now, the modality is upon modality is people are working. Raman or SERS modality can be coupled with the MRI, SERS modality coupled with the fluorescence, SERS modality coupled with the photoacoustic. So, this is an example of the MRI SERS modality which can be used for even it is also in the photothermal therapy. It is a gold nanostar, and which is attached with the silica cell, and here they used a gadolinium which is a potential agent for the MRI, and this is the DOTA, it is a ligand which is attached to that, and yes, it is a probe where this nanostar is attached with the DOTA gadolinium, and they have tuned in both ways. They can check the modality or progression through MRI and also in Raman. I am showing the Raman modality. In the tumor side, this is the tumor side, these 1 and 2 are the tumor side, where it is reflected the Raman signals on it, and these are the 2 regions throughout the non-tumorigenic regions, either skin and other sides. So, you may reflect this modality, and also that can be translated in the MRI. So, these are the experiments and these are the translations are people are working. So, it is called the photothermal evaluation that can be translated both ways. This is one of the another examples where nano vessel, where nano rod can be encapsulated in a vessels, and where you can use it as a theranostic probe, where targeted way you may use it, and these degradable plasmonic vessels can be used to as a theranostic probe for the cancer cells, and that can be here the chemotherapy or chemotherapeutic agent is coupled with this photothermal therapy. So, it is a chemophotothermal therapy that can be translated by this Raman's imaging or Raman spectral analysis. So, another example, if you induce the apoptosis through ROS, reactive oxygen species generations, and here the ROS generations can be done through hydrogen peroxide. So, here this is a platinum nanocube where the DNA damage can be reflected. This is the same work as like previous. DNA damage, only the substrate is different. DNA damage can be reflected in the Raman mode, and you may see the finest peaks which can be reflected in these 3 regions, and that can be translated, that ROS can induce apoptosis which can be reflected in the SERS mode using this substrate. This is very recent work in this area, where you know that folic acid used as a targeting ligand for many cancer cells and tissues is a nanostar which is also silica coated. Anti-folate antibody is attached. This is the non-targeted and this is the folate targeted things, and they have checked the real time what happened in the tumorigenic regions, and where they can tune in the liver, stomach, spleen and the tumor. So, all these areas they can translate the Raman imaging, and where they found that the targeted construct mostly localized in the tumor regions. So, these are the very recent work, and that tumor after this surgery they can take out and they can put it in a way to check all these organs, and found the mostly localized your nanoconstruct in the tumor side. This is one of the another finest work in few weeks before it is appeared in SES Nano, where you know that the tears, if you find out the arthritis and that thing, and if you check the percentage of uric acid in tears, that can be checked, that can be translated. Here they make a scale. The scale is made of the cellulose, and that can be used called the gold coated cellulose scales, and that morphology is like that. It is called the gold nano islands, and if you check the drop of steroids on it, you may easily translate it, the percentage of, even calculate the percentage of the uric acid on it, and you may think about what is the relevance and on this gout or arthritis diagnosis in the surface mode. So, these are the fine examples that people are translated, and it can be done very nicely with this spectroscopy. This is one of the finest way where the platinum electrodes are used for detection of this same way in the proteins, and this is also very recent work. This also modality of the Raman enhancement, and where mechanical trap means you may use as a trap, you may capture the cells, you may capture the analytes on the surface, and that can be translated, and you can check the protein and the lipid abundance on that trap. So, this is also used as a live cell or live cell imaging and also detection of these trapped molecules, especially the proteins and lipids on it. So, another finest example I am going to show you in for the especially the gastric cancer, two things. One is the early gastric cancer EGC and another is the advanced gastric cancers. Mostly people are doing the this volatile organic VOC, this component, where the nitric oxide and another volatile components can be analyzed through the GC. So, what they made the substrate, it is a graphene. If you use the graphene as a substrate and that graphene you may used as gold coated graphene as a substrate, where the surface area is quite huge, and if you just breathe on that substrate and subsequently if you can check its Raman band, then the VOC component can be analyzed, which can be equally analyzed in the GC, and for that you do the computational analysis and the percentage of these analytes can be translated through this computational analysis. So, maybe Satish will know much better on that aspect, but it can be translated and it is very recent work, and where they can nicely translated that AGC and EGC percentage in through this methodology. This is also very amazing work in the sense where if you translate these three modality, one is this MRI and the SERS, and that can be modulated in the tumor imaging, and where SERS, where you can see the normal and the cancer tissues with this silica coated Raman molecules, which is used as a Raman reporters. So, these are the nice examples where people can do extensively in the tissue samples and in many other aspects. Here there is a quantitative estimation of the nucleic acids. Mostly they have done the cytosine and variable component of the cytosine in the DNA in the nucleic base, that can be translated in the Raman mode using this substrate. So, it can be nowadays it can be translated what are the components of the nucleic acids, what is its percentage, that also people are working in semi quantitative and the quantitative ways. So, now I will give you some of the other examples apart from this cancer and these things. So, to check the environment pollutant, it can be utilized. How? Here is a gold nanorod and the iron oxide nanoparticles, which is combined together, and where this is the chip based, and with this it can be translated the check the environment pollutant, thyme and other things, and which can be taken as it is a magnetic nanoparticles. You can take out, first you make that as a tablet. So, if you use this tablet in a solutions of the what you want to check it in the either in aqueous or in solid, then pull out the tablet through the magnetic bar, and where the pollutant can be absorbed on it, and check with the handheld Raman, you may get the specific pollutant in very precisely. So, this is one of the recent work to check the environment pollutants. And this is another nice work. You know that two things happening, apoptosis and necrosis. So, how do you balance or how do you judge the apoptosis and the necrosis? So, that can be done through FACS, and this is PERS. PERS is called the Plasmon enhanced Raman spectroscopy. So, if you check in FACS analysis, you can see the viable apoptosis and necrosis percentage, and that same thing you may be translated, can be translated to this PERS, where you can see the difference in the spectral features that can easily translate these two things are complementary to each other. So, this is very recent work. where they can check the apoptotic and the necrotic factors through this modality. So, apart from working in this area, we first revealed some of the advancement in this modality in this review, and we found that these are the SERS, fluorescence, bioluminescence and photoacoustics. These are the imaging modalities. So, we try to evaluate its pros and cons. So, we just think about, yes, everything having some cons also. So, if you consider the SERS, definitely its sensitivity is quite huge, no way you can beat its sensitivity, but the limitation is that its penetration depth. So, it may be within this limit, but the fluorescence, it is relatively cheap, because that is why it is widely applied in biomedical applications imaging, but also it has the cons this one. Bioluminescence, the sensitivity is quite high, but difficulty in translation in clinics, that is still is a challenge. And photoacoustic, it is a very unique for in terms of penetration depth. So, we revealed all the pros and cons and its future in terms of this modality. So, I will show you one of the last example, where a brain surgery can be translated in 3 modalities. This is one of the unique work in Nature Medicine is published, where in the rat brain they have checked, they have analyzed, they have checked the progressions in MRI, Raman and the photoacoustic imaging probes, where they think about when the pre-surgery mode, if you incorporate the construct, which is a gold nanoparticle silica coated and the gadolinium is incorporated on it. So, in the pre-surgery to 1 week time in the post surgery, and if you turn these areas, you may find out some micron sized tumor you can locate, and that can be helped the progression of the surgery, and that can be easily translated. If you make a probe where it can be really correlated with the surgical unit and its progressions through this mode. So, they are saying that if you incorporate these 3 modality in a single probe, then it will be really nice in the futuristic development. So, this is, although it is a but a lot more works has been done in these areas, but people are trying to couple these 3 modalities in this fashion. Now, I will show you another 10-15 minutes what our contribution, especially in my group's contribution towards this advancement in Raman. So, we are the organic chemist. So, we are synthesizing molecules which should be the Raman actives, and we are trying to cover the whole range of this wavelength, and think about that we are specially interested sometimes in that near IR range, because we can do extensive in the in vivo work in tissue imaging. So, in this field, so we have synthesized some NIR molecules which is very Raman active NIR molecules, and these molecules can be translated in better way, and which will be the best Raman active molecules in terms of the signal intensity. So, that library synthesis we have turned up about 80 molecules, and out of which 1 or 2 molecules is really challenging, better than of the commercially available NIR Raman active molecules. So, we have done similar way the probes, Raman probes using gold nanoparticles, and that can be encapsulated with the biomaterials, and that can be conjugated with the target specific antibodies, and all things are characterized in terms of all physical modes, either tame and its stability. So, we try to translate these probes in first the cell imaging. So, if you this is the bright field image in the Raman microscope, and this is the Raman image, or Raman, it is called the Raman mapping. So, once it is the tagged, is targeted, these cells, if you target the cell surface proteins, then mostly the recognition in the cell surface. And if you see this mapped image in any box, if you click here, you get the corresponding the spectra on it. So, it is correlating the image and the spectra, where you can translate both the things together, that it is recognized. And that can be also done in the dark field image, where you can see the prominent scattered nanoparticles in the cell surface. So, that we have done in several cancer cell lines with the targeted imaging fashions. Also, we translated this one in the animal models, especially in the xenograft models, where the tumor area, the tags has been mostly localized after tailvein injections, it will be mostly localized in the tumor area and some of these other organs. But if you check from the tumor side the Raman spectra, you can get the corresponding spectra of that pure tags, which is resemble that it is localized, it is targeted to that site. We have done lot of molecules for the multiplexing studies, because if you have 3 or 4 analytes together, that you want to check the abundance or recognition of these 3 molecules, then you have to use this type of Raman active molecules, and each yellow shade is the corresponding signature peak for particular tags. So, that can be utilized for particular antibodies or targeting molecules, which can be checked for these particular areas. So, we have done even in the animals for the multiplexing studies, where 2 different antibodies or 2 same antibodies can be combined, and that can be translated, and that peaks can be correlated with these 3 tags. So, and also we have done lot of kinetics on it, because once this Raman tag, which is tagged with the particular antibody, it will be retained in the tumor site quite some time. It is almost 1 week time it is retained and slowly degraded. That study we have done in the live animals, and so that you can see that its efficacy or its effectiveness towards the tumor site. In recent years also we have synthesized some of the squarine based, this is called the squarine molecule, squarine based Raman reporter molecules, because our institute is pioneer in synthesizing lot of fluorescent molecules and Raman active molecules. We also working in the same line, and apart from this having the thiol containing molecules, which is having very nice and intense Raman peaks compared to that other 2, and that can be used for further or utilized for the diagnosis and detection modes. So, in the first cases we have checked in the 3 cancer cell lines, and we found that it will be really recognized if you have really tagged antibody on it, and these are the false color generated from that cell lines. So, each color, if you click it, you can get the corresponding spectra. So, if you have the tag with the corresponding antibody or corresponding protein or any other corresponding aptamers or nucleic acid which you want to target it, then you can easily recognize any type of specific cancer cells which is overexpressed in such type of proteins. In recent years also we did the work on cervical cancer detections, and here in cervical cancer we find the abundance of different proteins in the cell surface and the intercellular regions, especially in the nucleus, and we make the tags with 2 different types of antibodies, where you see one is the surface and one is the nucleus. So, when it is in the surface, the blue color represents the nanotag, means tag which is attached to the corresponding antibody, and the cyan color is the cytoplasm and the red is the nucleus. Actually, the cancer cell having huge nucleus. The difference in the cancer cells and the normal cells is that when there is a well grown cancer cell and a healthy normal cells, the nucleus of the well grown cancer cells is quite huge. They have almost nil cytoplasm. So, you can generate through this coding or false color here. So, it is that you can see the recognition pattern of this blue line, which is corresponding the surface recognitions, and you can see the blue in the nucleus region. So, it is the nuclear recognitions. So, two types of recognitions can be translated in cells in real time imaging, that can be done in this cervical cancers, and that also translated in what we did the immunostaining in the cervical cancer. So, that has been translated in the immunostaining process with the corresponding antibodies. So, as I talked about the therapy unit also. So, this is one of the fine example where we used chemo-photothermal therapy together, chemotherapy along with the photothermal therapy. Photothermal therapy coming through this nanorod, which is targeted to the MMP attached peptide sequence and also attached to the doxorubicin. In that case, we show that if it is a synergistic effect between chemotherapy and photothermal therapy, it will huge, and we can induce the apoptosis in a much better way. So, that phenomena we have integrated in a nanorod, which is the functionalized with the molecules, Raman active molecules, and these are the peptide sequence which is corresponding to the MMPs and also attached to the molecules, the doxorubicin, which generates or imparts the chemotherapy. So, that things can be translated in Raman. Why I told you some of the examples, they have shown that while you induce the apoptosis, the DNA backbone cleavage is occurred, and that can be checked in the real times. We show that the 833 peaks, which is corresponding to the DNA backbone, which is diminished immediately upon the laser irradiation, which occurs the photothermal therapy along with the chemotherapy. So, that can be validated through the DNA laddering experiment as well. In recent years, one of the recent development for the detection of the prostate cancers. Generally, the prostate cancers people are used to do in pathology the detection of the PSA level, and it has some limit for the PSA level, and mostly people are doing through the ELISA method. So, what we did here, we used a molecules and we used a peptide sequence which is specific to the PSA and which is conjugated with the nanoparticles. So, if you use the PSA enzyme, then this peptide sequence, mostly this amide linkage will be cleaved instantaneously, and once it is cleaved, then what happen, you see that this is the Raman active molecules, it will be far apart from the nanorod and its signal will be vanished. So, if you can change ratiometrically the intensity of the Raman signals over the concentration of the PSA enzyme, you may detect, you may even study what is the dose limit for the PSA enzymes. So, we found that we may come up the limit of detection even 0.5 nanogram with this modality. So, that we have tuned even in the cancer, prostate cancer cell lines. So, this is very recent work, and we are also trying more advanced way to explore this field in the surface enhanced Raman scattering, to especially on the detection in different analytes in the cancers. And very recently we try to develop one level free detections modality, especially in the screening of the cervical cancers. So, this is our very recent ongoing work. Generally, the cervical cancer people are in the pathology, people are used to do the pap smear test, and which is very conclusive, and which people are also telling that it is very subjective also. But the problem is that in many cases you need to come up very good conclusion on the stages of these cervical cancers, which requires repeated time of the pap smear and which is sometimes very confusing. So, we try to develop in a level free method only using the nanoparticle as a substrate, what are the changes in the biochemical modes in the cervical cancers. Mostly if you see the cervical cancer, there are quite a few number of stages, but in broadly speaking, if you categorize these three stages, one is the normal, one is the HSIL, which is the in between the normal and the fully carcinoma stages, and this is the fully carcinoma stage. So, we try to evaluate the spectral features of the proteins, amino acids and lipids in these three stages, and we try to validate these things with first even the cell lines, then lot of cervical exfoliated cells from the patients, and that work almost we are validation part we have done. So, we found the specific bands which is present intensely in the HSIL, or it can be present intensely in the carcinoma, but it may not be present so intense in the normals. So, that small reflections can be correlated in statistical calculations that we are doing with the help of Dr. Sotis here, and we try to validate more way that if you have the spectral pattern, then we can say which selective way or which how much selectivity or specificity on that patterns. So, that may be a come up a good alternative if we can establish this validation part in these 3 regions. So, with this I like to stop here, and we are having lot more ongoing works, but due to the time limitations I like to stop here all this advancement in this field, but there definitely a future perspective in this field if we can work more and try to develop more. So, I come across all these things starting from what is SERS evaluations, biomarkers, and what is the modality of the therapy, and what is its relevance, and what is its future perspective. Maybe in future people working in this field can come up a SERS based endoscope, which definitely alternative mode for many disease diagnosis and detections. So, this is before that I would like to end, I would like to acknowledge all my peoples. With this I stand here and this one, and these are the most even our collaborators who helped me to come up this result, and the funding agencies, and definitely with all support of my students I am here, and 2 of my students are here participating this, Varsha and Dr. Monu is here, and they are contributing a lot to come up this stage. Thank you very much for your kind attention.
Thank you, sir. The topic is open for discussion. Sir.
What is the Raman activity about copper nanoparticles?
Copper has been tested in few cases, but it is not a good Raman substrate or SERS substrate. It can be used as a substrate, okay, like gold, silver, copper also used as a Raman substrate, but somehow it found that in these 2 mechanisms, electromagnetic enhancement and the chemical enhancement is not so impressive like gold or silver. So, it may be used, but it may not flourish.
When these particles come in cluster size, gold nanocluster, copper nanocluster, will it be Raman active?
Yeah. Raman activity as a if you use as a substrate, that definitely depends on its size and shape, okay, mostly size. So, it has been found that in case of the gold, if it is a cluster means it is maybe 2 nanometer or 3 nanometer size, it may not be Raman active. So, if you tune the size in terms of around 20 to 50 nanometer range, you can get maximum enhancement, because the surface area is one of the key factor for getting the good enhancement. So, cluster definitely may not be a good substrate.
What type of laser is used for?
Yeah.
What is the power of the laser?
Yeah, there are as I said in the basic principles, we can use the laser from the UV to NIR range, but it is a single laser generally people are using for the Raman active. It starts from 488 to 1100 something, but it is a single laser 488, 532, 633, 785, 835 is also available, and 1100 also something is available.
So, in the case of
Huh?
In the single
Yeah, single instrument you can couple 2, 3, 4 lasers together.
commercially available.
Yeah. In our institute we used to use having 2 lasers, one is 633 and another is the NIR.
What is the cost?
Total system or the laser?
Laser.
See, it varies. Maybe if you buy some of the good components like Vitek from Germany, we have that machine. Each laser cost maybe around 30,000 to 35,000 US dollar.
Then in one of the slides, star-shaped golden nanoparticles are there. And any advantage is there with this particular shape?
In the star shape, it has lot of spikes. Maybe you can control the spikes 3, 4, 5, 6. Once the number of spikes are increased, then two things may happen. One, its surface area may be increased, and if you incorporate some of the Raman active molecules on it, you may generate more number of hotspots. Hotspots means the surface and the molecules in between the closed gap, you can tune it. Okay. If the closed gap is very close within 0.5 to 1 nanometer, then electromagnetic enhancement will be quite increased, and also if you have the molecules which having the chemical enhancement, which is secondary things. So, if it is star shape, you may think better enhancement because of these two factors.
Then in one of the slide, MRI is there.
Yeah.
That is FP3 incorporated slide.
Hmm.
And any use is there with this gold nanoparticle in MRI?
No, gold nanoparticle, nothing related to the MRI gold nanoparticle.
Simultaneous imaging.
Yeah, but gold nanoparticle is used for the surface enhanced Raman, but iron oxide which is used as a either coating agent on it, that can be used for the MRI. So, you can do simultaneously both MRI and the Raman.
Okay, in one of the slide, that is multi-model imaging, silica shell is there in the outside of Raman active layer.
Hmm.
And is this silica layer will weaken this SERS signal?
No, 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 no. Silica used actually the silica used in different ways. It is used to increase the stability of the molecules. Silica used to incorporate some of the molecules you intend to incorporate, because it has a, if it is a mesoporous silica, then you can incorporate in the pores, or you can functionalize some of other functionality. So, it is used for to make other functions to in order to make it.
So now TERS is also there.
Yeah, TIFF enhanced Raman spectroscopy.
What
Yeah.
is the advantage of TERS?
In TIFF enhanced, you may get more precise detection, okay, and you may get more precisely located area which you want to detect, because in TIFF enhanced your substrate in the tip. Okay. So, that in a reverse way, your substrate in the tip, your analyte in the base. Okay. So, that you can focus more precisely and to the substrate, then it has a chance for quantitative detection. So, TIFF enhanced also coming up, or it also having some of the disadvantage over normal surface enhanced, but TIFF enhanced also coming up, more precise detection basically.
Other than the helium neon laser, which is the one you are using?
We are using also some cases solid state laser and nitrogen laser also.
Which
We are using 633 and 785, 532 also available. In this area, Suchitra having confocal Raman, they have 2 laser 532. BSCC also having Raman, they are also using 532 and 785, and we have also.
Thank you.
Thank you, Scott.
You're welcome.